Dear students, today we will discuss about primary productivity and factors affecting primary productivity. The main objectives of today's module are to help you understand the basics of primary productivity, how it helps to know about the factors affecting primary productivity, to make you understand factors affecting primary productivity. First of all, brief introduction. Plant production is defined as the amount of energy available to sustain all organisms, including humans. We depend on plant production directly for food and fiber and indirectly because of the critical role of vegetation in all ecosystem processes. Now, let's discuss primary productivity and various terms related to it. The rate at which radiant energy is converted by photosynthetic and chemosynthetic autotrophs to organic substances is termed as primary productivity because it is the first and basic form of energy storage. However, the total rate of photosynthesis or energy assimilated by the autotrophs is termed as gross primary productivity, and the rate of energy storage as organic matter after respiration is termed as net primary productivity (NPP). NPP can hence be described by the following equation. So, net primary productivity is equal to gross primary productivity minus respiration due to autotrophs. That is, NPP is equal to GPP minus R. So, NPP is the balance between the carbon gained by gross primary production and carbon released at the ecosystem scale, usually over relatively long time intervals, such as a year. Basically, these are those organic compounds that diffuse or are secreted by roots into the soil, root exudation. The carbon transfers to microbes that are symbiotically associated with roots. For example, mycorrhizae and nitrogen-fixing bacteria, and the volatile emissions that are lost from leaves to the atmosphere. Productivity is usually expressed in units of energy per unit area per unit time or kilocalories per square meter per year. That is kcal per meter square per year. However, productivity may also be expressed in units of dry organic matter, that is gram per meter square per year. Let's come to know how to measure net primary productivity. The simplest and most common method of measuring net primary productivity in terrestrial ecosystems is to estimate the change in standing crop biomass (SCB) over a given time interval. T2 minus T1 minus SCB is equal to SCB into T2 minus SCB into T1. Two possible losses of biomass over the time period must also be recognized. That is, loss of biomass due to the death of plants, that is D, and loss of biomass due to consumption by consumer organisms, that is C. The estimate of net primary productivity is then NPP is equal to SCB plus D plus C. In aquatic ecosystems, the most common method of estimating NPP is the light-dark bottle method because oxygen is one of the most easily measured products of both photosynthesis and respiration. Hence, a good way to gauge primary productivity in an aquatic ecosystem is to measure the concentration of dissolved oxygen. Let's discuss the experiment. In one set of clear glass light bottles, a water sample from the aquatic ecosystem and associated autotrophic organisms, phytoplankton, is allowed to incubate for a defined time period. If photosynthesis is greater than respiration, oxygen will accumulate in the water, providing an estimate of NPP. On the other hand, water is also incubated over the same time period in another set of dark bottles, painted dark to prevent light from reaching the water. Since the lack of light will prevent photosynthesis, hence the oxygen content of the water will decline as a function of respiration. The difference between the values of oxygen in the light, that is photosynthesis minus respiration, and dark, that is respiration. Bottles at the end of the time period therefore provides an estimate of total photosynthesis or gross primary productivity. Now let's discuss the various factors affecting primary productivity in terrestrial ecosystems. Sites with a higher mean annual temperature typically support higher rates of photosynthesis and are associated with a longer time period over which photosynthesis can occur. At a global scale, the largest ecosystem differences in NPP are associated with variation in climate. NPP is greatest in warm, moist environments. NPP is greatest in warm, moist environments where tropical rainforests occur and is least in climates that are dry, for example deserts or cold. When dry ecosystems are excluded, NPP also increases exponentially with increasing temperature. The largest differences in NPP reflect biome differences in both climate and vegetation structure. Dear students, there is another factor that affects primary productivity, that is precipitation. The higher the rainfall, the more water is available for transpiration. This amount of water available to the plants will therefore limit both the rate of photosynthesis and the amount of leaves, that is surface area that is transpiring, that can be supported. Although there is independent effects of temperature and precipitation on primary productivity, but in reality the influence of these two factors is closely related. This can be explained by the following phenomenon. As air temperature increases, the potential for evaporation increases and therefore increases the rates of transpiration and plant water demand. Now, if temperatures are warm but water availability is low, productivity will also be low. Conversely, if temperatures are low, rates of photosynthesis and productivity will be low regardless of the availability of water. Hence, it is the combination of warm temperatures and an adequate water supply for transpiration that gives the highest primary productivity. In addition to the above mentioned factors, there is also influence of nutrients on primary productivity. The availability of essential nutrients required for plant growth directly affects ecosystem productivity. The availability of nutrients in the soil influences the rate of nutrient uptake, photosynthesis, and plant growth, which results in a general pattern of increasing NPP with increasing soil nutrient availability. It is because of the environmental controls over photosynthesis, for example, light and water availability, that strongly influence photosynthetic carbon gain. However, on weekly to annual time scales, plants appear to adjust leaf area and photosynthetic capacity so that carbon gain matches the soil resources that are then available to support plant growth. Plant carbohydrate concentrations are usually lowest when environmental conditions favor rapid growth. That is, carbohydrates are drawn down by growth and tend to accumulate during periods of drought or nutrient stress or when low temperature constraints. NPP. You might be surprised to know that tropical forests typically have higher NPP than other biomes. It is because of the litter production in these forests that tend to correlate with the supply of nutrients, especially phosphorus, suggesting that NPP in tropical forests may also be limited by the supply of below-ground resources. NPP in tropical forests is maximal at intermediate levels of precipitation. A study has confirmed that NPP in dry tropical forests is moisture limited, but in extremely wet climates, NPP declines in response to increasing precipitation, probably due to oxygen limitation to roots and or soil microbes and in leaching loss of essential nutrients. NPP in tropical forests is therefore probably limited by the supply of below-ground resources, including nutrients and also due to below-ground water or oxygen. Well, apart from tropics, in a temperate salt marsh, where water and nutrients are abundant, NPP responds directly to increases in carbon dioxide, as do crops that receive a high nutrient supply. However, NPP is enhanced by nutrient additions even in the most fertile agriculture systems, indicating the widespread occurrence of nutrient limitation to NPP. As we already know that globe comprises of terrestrial ecosystem and aquatic ecosystems, and have already discussed factors affecting primary productivity in terrestrial ecosystem. Now let's have a look on factors affecting primary productivity in aquatic ecosystem. Since photosynthetically active radiation PAR declines exponentially with water depth, hence, the photosynthetic rate and subsequently the gross productivity of phytoplankton are highest at intermediate levels of PAR. On the other hand, the respiration rate does not change much with depth. This means that as the phytoplankton goes deeper in the water column, the photosynthetic rate declines as the light intensity decreases until at some point the rate of photosynthesis, that is gross production, is equal to the rate of respiration and net primary productivity NPP is 0. This zone is referred to as the compensation depth and corresponds to the depth at which the availability of light is equal to the light compensation point. In the oceans, nutrients in the deeper waters must be transported to the surface waters where light, that's PAR, is sufficient to support photosynthesis. As a result, nutrients, particularly nitrogen, phosphorus and iron, are a major limitation on primary productivity in the oceans. Let's see what John Downing, an ecologist, examined from more than 300 nutrient enrichment experiments conducted in marine habitats around the world. He found that nitrogen addition stimulated phytoplankton growth the greatest, followed closely by the addition of iron. In contrast, the addition of phosphorus did not, on average, stimulate phytoplankton growth. These results confirmed that nitrogen and iron are the two most limiting nutrients in marine environments. A map has been presented which shows that the most productive waters of the oceans are the shallow waters of the coastal environments. This happens for two reasons. First, shallow waters allow for a greater transport of nutrients from the bottom sediments to the surface waters, aided by wave and the changing tides. Second, coastal waters receive a large input of nutrients carried from terrestrial ecosystems by rivers and streams. Now let's understand the role of organic carbon on productivity of aquatic ecosystem. Organic carbon produced within an ecosystem is termed as autochthonous, while as inputs from outside the ecosystem are termed as allochthonous in aquatic ecosystems. In aquatic ecosystems, the allochthonous input is through the photosynthesis by aquatic plants, attached algae in the shallow waters and by phytoplankton in the open waters. In contrast, small stream ecosystems flowing through forested catchments derive most of their organic carbon from dead plant materials deposited into the water from the surrounding forest ecosystem. Have a look on the significance of above-mentioned sources of carbon. In most marine ecosystems, autochthonous inputs of organic carbon dominate due to the high net primary productivity of resident populations of phytoplankton, whereas small stream ecosystems flowing through forested catchments derive most of their organic carbon from dead plant materials deposited into the water from the surrounding forest ecosystem. Shading from trees limits available light in the waters, preventing any significant growth of phytoplankton or attached algae. In these streams, allochthonous carbon provides energy resources that support levels of consumer abundance beyond those that could be supported from autochthonous sources. As the stream widens further downstream, the shading by adjacent trees becomes limited to the stream margins, and the increase in available light in the water column allows for an increase in net primary productivity by submerged aquatic plants, algae and phytoplankton. The result is a continuum of the relative importance of allochthonous and autochthonous sources of carbon in the energy balance of flowing water ecosystems from the smaller headwater streams to larger river systems. Dear students, if there are factors affecting primary productivity, the converse may be also true, like energy allocation and plant life form that influence primary production. This may be answer to your query as why there is pattern of decreasing net primary productivity, that is NPP, with declining precipitation. Reduced moisture conditions result in an increased allocation of carbon to roots at the expense of leaves, thus reducing leaf area and rates of net carbon gain. A high leaf area is necessary to maximize NPP, yet the major factors that constrain NPP are below-ground resources. The plant faces a dilemma of how to distribute biomass between leaves to maximize carbon gain and roots to maximize acquisition of below-ground resources, and for that matter, plants exhibit a consistent pattern of allocation, that is, the distribution of growth among plant parts that maximizes growth in response to the balance between above-ground and below-ground resource supply rates. Plants allocate new biomass preferentially to roots when water or nutrients limit growth, and new biomass preferentially to shoots when light is limiting. Plant hence can increase acquisition of a resource by producing more biomass of the appropriate tissue, by increasing the activity of each unit of biomass, or by retaining the biomass for a longer time, thereby increasing NPP. Do you know how many strategies a plant adopts to enhance NPP? For example, it increases carbon gain by increasing leaf area or photosynthetic rate per unit leaf area, or by retaining the leaves for a longer time before they are shed. Similarly, a plant can increase nitrogen uptake by altering root morphology or by increasing root biomass, root longevity, nitrogen uptake, rate per unit root, or extent of mycorrhizal colonization. NPP in most ecosystems responds most strongly to a particular resource, for example, to water in deserts, grasslands, and arid shrublands, to nitrogen in tundra, boreal forests, and temperate forests, and to phosphorus in tropical wet and dry forests. Thus, as a first approximation, deserts are water-limited ecosystems and temperate forests are nitrogen-limited ecosystems. In many ecosystems, NPP responds to increased availability of more than one resource. Why does this occur? The simplest view of environmental limitation is that growth is limited by a single resource at any moment in time. Another resource becomes limiting only when the supply of first resource increases above the points of limitation. That is Liebig's law of the minimum. At least 4 processes contribute to the multiple resource limitation observed in many ecosystems. First, plants adjust resource acquisition to maximize capture of the most limiting resource. Second, changes in the environment, for example, rain, storms, pulses of nutrient supply, change the relative abundance of resources. Third, plants exhibit mechanisms that increase the supply of the most limiting resource. Fourth, different resources limit different species in an ecosystem. So, ecosystem scale NPP responds to the addition of more than one resource. Plants adjust resource acquisition to maximize capture of and minimize limitation by the most limiting resource. As I have already discussed earlier, plants adjust allocation of new production to roots versus shoots to minimize limitation by below ground versus above ground resources respectively. Plants also alter allocation within the root system to maximize capture of the most limiting below ground resource. In deserts, for example, nutrient availability is greatest close to the soil surface, whereas water supplies are generally more consistently available at depth. As a result, the amount of nutrient or water that a new root acquires depends on the depth at which it is produced. To acquire water, some desert plants produce coarse, deep water roots which effectively conduct water. but are relatively ineffective in absorbing nutrients. Other plants produce only shallow roots and remain active only when surface water is available. In addition, the biochemical investment by roots is specific for each nutrient. For example, nitrogen uptake requires synthesis of specific enzymes to absorb nitrogen, reduce nitrate, and assimilate reduced nitrogen into amino acids, whereas different enzymes are required to absorb phosphorus. On the other hand, changes in the environment, for example, rain, storms, pulses of nutrient supply, change the relative abundance of resources, so different factors limit NPP at different times. It must be also known that temporal changes also limit NPP because essential resources do not become equally available at the same time. Many ecosystems experience a pulse of nutrient availability at the beginning of the growing season, when temperatures may be suboptimal for growth, thereby showing enhanced NPP. Meanwhile, temporal changes in the process of limitation of NPP are buffered by storage. Plants accumulate carbohydrates or nutrients during times when their availability is high and use their stores to support growth when the supply declines. Over seasonal timescales, plants use stored carbohydrates and nutrients to support their burst of spring growth and replenish these stores at other times when photosynthesis and nutrient uptake is less. To sum up, storage enables plants to acquire resources when they are not readily available and use them at times of low supply, thus reducing temporal variation in the nature of resource limitation. It's useful to know that plants exhibit several mechanisms that increase the supply of the most limiting resources. Plants having symbiotic associations with nitrogen-fixing microbes directly promote nitrogen inputs to ecosystems. Some actomycorrhizal associates of plant species break down proteins and transport the resulting amino acids to plants. Some plants enhance the supply of phosphorus through the production of organic chelates that solubilize mineral phosphorus or through the production of phosphatases that cleave organic phosphates. Plants also exude carbohydrates that enhance mineralization near the roots. Different resources limit different species, so ecosystem scale NPP responds to the addition of more than one resource. Every species in an ecosystem has slightly different environmental requirements and therefore will be limited by different resource combinations. Tundra species, for example, differ in their response to temperature, light, and nutrients, and in some cases to the addition of nitrogen versus phosphorus. These differences among plant species in the factors limiting growth contribute to the coexistence of species in a variable environment. This may be particularly important in explaining why species differ in their productivity among years and why the productivity of ecosystems varies less among years than does the productivity of any of the component species. Spatial heterogeneity in the supply of potentially limiting resources is another important reason why different plants may be limited by different resources. Many organisms adjust their physiology and behavior based on innate circadian about 24-hour rhythms that lead to 24-hour cycles. For example, stomatal conductance and carbon gain show a circadian rhythm even under constant conditions because stomata have an innate 24-hour cycle of stomatal opening and closing. Plants store starch in the leaves during the day and break it down at night, so the rate of carbohydrate transport to roots is nearly constant. This was all about today's lecture. Hope you have understood it well. Thanks.